complete damage of brain organs. So for Parkinson disease, the causative factor is alpha synuclein protein that can form aggregates in brain organ. Okay, not the last. The previous one is not the last. Okay. So then we look at the type 2 diabetes. It involves a myelin protein. This kind of protein is produced by beta cells in pancreas organ. It is also a kind of hormone that can modulate the metabolisms in human body system. Similar to previous diseases, myelin can form fibrils and aggregates and subsequently can cause damage on beta cells. When the beta cells are damaged, diabetes mellitus develop. Okay, moving on to an illustration to show how aggregation is formed. At the beginning, proteins are folded into correct 3D structure. Okay? And due to certain condition, the proteins become incorrectly folded, and subsequently the formation of aggregates or oligomer. Continuous aggregation of this protein lead to formation of fibrils. Okay, so it looks like a long cylinders structures, and combination between more than one fibrils can lead to the problems in brain organ. However, all these steps they are actually reversible if the relevant therapy is given. In the case of incorrectly or misfolded protein happens, it can still be controlled through degradation process. If it is not properly controlled, it leads to formation of fibril and subsequently aggregates. Okay, type 2 diabetes, which is different from type 1 diabetes. So here we can see a myelin protein, tau protein, insulin, and others. They are interacting, causing a problem in brain organ. Okay. Okay, we finish discussion on different diseases due to the problem in the problems like mutation and also abnormal protein structure. Okay, now we move to biomarkers discussion. As I mentioned just now, biomarkers refer to a set of functional proteins in cellular system. They are used to monitor the normal function, to monitor health, to monitor disease development, and others. And there must be some characteristics of biomarkers. First, for example, it should be specific enough. It can be monitored in certain disease only, but not in other disease. Specificity of biomarkers can help medical scientists to produce analysis to monitor the disease development with high sensitivity. So as shown here, at least we have 5 characteristics of biomarker. Uh, for example, they are minimally invasive, inexpensive. I mean, we can use a simple method to detect the biomarkers in any biological samples like serum and other body fluids secreted by disease tissue only. This is what I mentioned about the specificity, highly sensitive, and yeah, highly specific as well. So these are the important characteristics of a biomarker. In this case, biomarkers refer to a set of proteins that can be used to monitor disease. We do not look at the use of DNA or other biomolecule as biomarker here. We focus on the proteins. Okay, different types of biomarkers. For example, translation biomarker and disease biomarker. So disease biomarker is about the one that can relate to clinical outcome or measure of disease. Okay. Other type of biomarkers include efficacy biomarker, mechanism biomarker, and target biomarker, and others. So you can read on your own. So basically, they share the same characteristics. They must be specifically represent the certain stage of disease or mechanisms. Okay, proteins as biomarkers. So we can obtain the proteins from serum, from urine samples, from other body fluid as well. So we can monitor specific disease based on these biomarkers. So in this case, we are discussing about uh, protein biomarkers to monitor disease in humans. So we are not discussing about biomarkers for uh, plant disease or other abnormalities. We focus on biomarkers in human body system. So the body samples, the samples like human serum can be used for analysis, such as SDS and subsequently MS analysis. So MS analysis is to determine the sequence of amino acid based on the mass spectra. And comparing the mass spectra between normal and abnormal samples can help us in identifying, can help us in diagnosing disease, and also understand the current stage of disease. For example, we compare a cancer patient and a normal or healthy individual. Okay, taking their serum samples, then analyzing using MS analysis, we can compare their protein profiles based on mass spec.